Well, this lecture is very near and dear to my heart. This is actually my distinct passion in all of this biology and genetics fields that we've been talking about all this time. This lecture actually doesn't come from any one chapter in our textbook. This is a kind of a conglomeration of a lot of different topics that I've put together over the years, and I'm really just trying to give you an overview of how DNA works and what DNA's role is in the living cell. You see, everything we go on to do after this lecture, starting with gene mutations and then the control of gene expression in bacterial cells, do a little bit of quantitative genetics and end the course with population genetics, all of that is going to require at least some understanding of DNA as a molecule and how DNA behaves as a molecule encoding proteins. So it's critically important right now at this point in the course to take a step back and discuss more the molecular aspects of DNA and how DNA is the instruction manuals for building proteins, which is the central dogma of molecular biology. So if there are some details in today's lecture that seem a little bit too in-depth for you or start to confuse you, feel free to just kind of bypass those details. Come to me, make sure that you're not missing any core concepts, but we're much more interested in the big picture in this lecture. So we're going to start off talking about the chemical nature of DNA and RNA. These are, after all, chemical molecules. And so to understand how DNA and RNA behave as genetic molecules, we first really need to understand their chemistry. We'll then move on to the structure of DNA and RNA and get an idea of how these molecules look and how that structure allows them to have the function that they do. From there, we'll transit to the idea of the central dogma itself, talk about how proteins are made. We'll talk a bit about transcription and just a touch more on translation, getting us all the way to protein synthesis. And then at the very end, just with pictures, at the very end on a single slide, we'll touch on the fact that proteins actually fold up, and that's what gives proteins their function. So for much of the semester so far, we've been discussing how Mendel's concepts of genetics and heritable traits, and then the expansion on his ideas of heredity and genetics, relates to molecular biology. We've said that traits are encoded in DNA. We've said that many times. We've defined DNA as being molecules that are divided into individual genes. I've told you a number of times already that these genes are the instructions for how to build proteins, and I can't say it enough, proteins are the machines of the cell. Things get done in the cell by proteins. Proteins are the doers of the cell. So we've said all this, but I don't think we've ever really tied it together intentionally as one synergistic concept. So that's what we're going to do today. We know already that there are two forms of genetic molecules. There are two ways of containing genetic information in a living cell. They are, of course, DNA, or deoxyribonucleic acid, and RNA, its close cousin, ribonucleic acid. DNA is ideal as a storage molecule. All of the information for building and regulating all of the proteins in our cells are stored in DNA. This means that the information for being you, because the proteins are what made you you, all of the information for being you is also contained in the DNA. So in every single one of your cells, because every single one of your cells has an identical genome, the DNA is identical in all of your cells except your gametes, in every single one of your cells is the information for being a skin cell, a liver cell, a lung cell, a neuron, etc. What do I mean by saying the information for being these cells is there? Well, what I mean is the information for building the proteins that a skin cell needs is in every single one of your cell types. The information for how to build the proteins that a liver cell needs, because liver cells need different machines, need different proteins than skin cells do, the information for building those proteins are in every single one of your cells. The information for building lung cell proteins are in every single one of your cells. So the information for how to build these proteins exists in every single one of your cells. How does a skin cell become a skin cell, a liver cell become a liver cell, and a neuron become a neuron? Well, the skin cell expresses only skin cell genes. That is to say, the skin cell only makes the proteins that the skin cell genes encode. The liver cell doesn't make any of those skin cell proteins. The liver cell only makes liver cell proteins. This implies that there must be regulation of genes, and in fact, of course, there is. Some genes are turned on so that proteins are made from them, while other genes are kept off so that proteins cannot be made from them. And this is how we can have identical genomes in every cell, but yet very different protein compositions in those different cells as well. So to get there, To allow this DNA to see to the building of a protein, we need an intermediate, and RNA is that intermediate. RNA is a working copy of the genetic information that was on the DNA gene. Proteins are made from genes found in the DNA. Yes, they are. However, RNA is the middleman. So the central dogma really is, to jump the gun here, DNA is copied into RNA, and the RNA is what's used to make the protein. But again, the information came initially from the DNA. DNA is the storage molecule for that information. So we're going to cover quite a few concepts today, because now we're going to slowly build the story from a DNA molecule, which we might not even be completely familiar with, even at this point in the course, all the way to a built protein. So here's how I like to think of the flow of information, or the individual roles that DNA and RNA play in all of this. You can think of the genome, the DNA, as the encyclopedia of life. Most people don't use encyclopedia anymore, but you may be familiar with the concept. Encyclopedia are big. My father had one in his office when I was growing up. Office. It was a small room off of the dining room, but he had a set of this in a bookcase, Encyclopedia Britannica, and it was many, many volumes, each volume very, very heavy. It was a lot of information. And that's what your genome is, a lot of information encoded on lots and lots of DNA, 6 feet of DNA in every cell to be exact. So let's say I come to you, you have an encyclopedia in your house, and I come to you and I tell you I need to know some information about a town called Wrexham. This town is a town in Great Britain. And you say to me, well, come on in, I can help you. I have an encyclopedia, I can help you with that. Here you go. And you hand me this entire set of encyclopedia. That's how you're helping me. And you say, somewhere in here is information on the town of Wrexham. Help yourself. You know, I'd say thanks, but no thanks. That's a little too heavy and a little too cumbersome. Never mind. But if you said, oh, sure, come on in. I have an encyclopedia. Let me look up Wrexham for you and Xerox that page. And you can take this page with you and use it for your report. I'll copy that information on Wrexham onto a smaller, more portable, more usable format for you. I would be very, very grateful. I'd thank you very much, and I'd go on my merry way with my copy of Wrexham. DNA is the set of encyclopedia. DNA is storing all of that genetic information in the nucleus. So when you knock on DNA's door and you say, hey, I'm trying to make this one protein, can I look at your encyclopedia? Can you show me the instructions for building that protein? The nucleus could hand you the entire genome and say, yeah, somewhere in this 46 chromosomes is the information you're looking for. Help yourself. And you would say, thanks, but no thanks. That's a lot of DNA, and I just don't have the time. But if there were a mechanism to say, oh, of course, why don't you come in? I'll Xerox that information on the one gene you're interested in. I'll make a portable, smaller, usable copy of that information. And you can have that. You'd thank the DNA very much, thank the nucleus very much, and be on your merry way. So DNA is everything. DNA is all of the information for building all of the proteins, but that's often too much information. It's just too bulky. RNA is that single Xeroxed copy. RNA is small, it's usable, it's portable. It's a copy of that one gene that you needed at that one time. It's the single piece of information that the cell requires in that moment. And so there's a role for each of these. We need a central repository for all the information. Of course, we need that. We need the DNA. We need all of the information to be localized in one place. But as we need to access that information, we don't just want it stored. We need to access it, manifest that information. We have to have some mechanism to turn that huge repository of information into a copy that we can actually use. So before we talk about how DNA and RNA play a role in making proteins, we really have to have a clear understanding of what DNA and RNA are as molecules. And in brief, DNA and RNA are chains. You'd more accurately refer to these as linear polymers, but they are chains. Chains of repeating subunits, chains of repeating small molecules, and for the case of DNA and RNA, those molecules are nucleotides. So DNA and RNA is nothing but a chain of nucleotides strung together one after the other. There are 4 different nucleotides used in DNA and RNA. Adenines, abbreviated A, thymines for DNA, but uracils take the place of thymine in RNA, guanines, G, and cytosines, C. Now here's the critical part. These nucleotides themselves do not contain any information at all. It is the order of these nucleotides that determines or contains the information that DNA holds. It is the order or sequence of these nucleotides that carries the information. So the instructions for building a protein are encoded in genes, but is the order of the nucleotides in those genes that contains that information. So the order of things containing information, small things, containing very little to no information on their own, but the order or sequence of those things does contain information. That makes me think about something. Are we already familiar with something that works this way? Here's some words for you. Tothcarly dandimad. That mean anything to you? Doesn't mean anything to me. But if I say the information is not in the individual letters, if I say the information is in the order of these letters, then perhaps if I reorder this same information, meaning will come out. Ah, my daughters, I get it now. It's the order or sequence of the letters that contains the information, not the individual letters themselves. The individual letters are needed to create the sequence, but it's the sequence that matters. And it works exactly the same way for DNA and RNA. The individual nucleotides are necessary, of course, just like we need our letters. But it's the sequences, how these nucleotides are strung together that carries the information. So you can very much think of the 4 nucleotides of DNA and RNA being the alphabet of life. Each individual nucleotide contains no inherent information on its own, but strings of nucleotides in a specific sequence contain the information for building proteins. And our millions of genes across all life forms on this planet contains the information for building millions of proteins. Just as strings of letters can make millions of words, and strings of notes can make millions of songs, strings of nucleotides make millions of proteins. It's all about the sequence. It's all about the sequence. So that's how the DNA and RNA molecules are similar. Are there differences? Yeah, there are a few kind of fine point differences. We'll talk about one now, and we'll touch on the other 2 as we go further on in the lecture. One of those differences lies in the structure of the nucleotides themselves, specifically in the sugar part of the nucleotide. Nucleotides are made up of 3 parts. We'll come back to this in just a moment, but they have a phosphate group, a sugar group, and the base, which actually gives it its identity, A, G, C, or T. The names of DNA and RNA actually say it all. Deoxyribonucleic acid, ribonucleic acid. That ribose sugar, the sugar part of the DNA nucleotides, have been deoxified. That's not really a word, but that's what happened. Here is the ribose sugar, the sugar component of a nucleotide for RNA. Here is the ribose sugar, the sugar component of a nucleotide for DNA. Notice that this ribose sugar for RNA, has 2 hydroxyl groups. Has 1, 2 hydroxyl groups. But for DNA, one of those hydroxyl groups has been deoxified. The oxygen has been taken away, and we're left with a single proton. So deoxyribonucleic acid is telling us that the oxygen of this hydroxyl has been removed, deoxy. And there are structural implications for that. That causes DNA and RNA to behave slightly differently as molecules. I won't go into it because it's not a chemistry class, but DNA is a bit more stable than RNA because of this, among other things. So that's one of our differences. But regardless, whether we're dealing with DNA or RNA, if we're going to take these individual nucleotides, these individual letters, and build genes with them, string them together into specific sequences, we need a physical way by which we can chemically link individual nucleotides together to create these chains. So we've got to take this nucleotide, which again is made up of a phosphate group, those ribose sugars, and then the nitrogenous base, the base, and this base is going to be either an adenine base, a thymine base, a guanine base, or a cytosine base. So all nucleotides are the same in their phosphate and their sugar. It's only the base that gives them their identity, their differences. We need to string these together to make a chain. Remember, it's the chain or sequence of nucleotides that contains the information for building proteins. That's what's important. So how do we link nucleotides together? How do we make these chains? Well, the chemistry is a little bit beyond this class. I'm not going to go into the hardcore bond formation. We'll do that in another class later on, and that was also covered in biochemistry. But I will tell you that the bond that's created that holds or glues the phosphate of one nucleotide to the hydroxyl group of the nucleotide before it, that bond is called a phosphodiester bond. So phosphodiester bonds hold nucleotides together into chains. Also, please note in this diagram here, as well as in this diagram here, the phosphate group is shown as negatively charged. And indeed, that is the case. DNA is a negatively charged molecule. And even that has lots of applications for how DNA can be used by the cell and how that negative charge is exploited. But that, too, won't be talked about until a little bit later this semester, just a touch, and then we go into that in some depth in cell and molecular biology. So DNA and RNA share three nitrogenous bases in common. Both DNA and RNA have adenine, cytosine, and guanine bases, again abbreviated A, C, and G. It's that fourth base where they differ. DNA uses thiamine as its fourth base. The fourth base of RNA is uracil. Thiamine and uracil are extremely close in their structure, very, very similar molecules, but different in very small and subtle ways. So we have four bases that make up our double-stranded DNA, guanine, cytosine, adenine, and thiamine. Cytosine, guanine, and adenine also present in RNA, but uracil is the fourth base there. We say that T's and U's, or thiamines and uracils, are synonymous, meaning they mean roughly the same thing in their information content, and they're somewhat substitutable for each other. They're structurally and functionally very similar. And please note that I did share with you the second difference between DNA and RNA. DNA has thiamine and RNA has uracil. The first difference was that the ribose sugars of RNA have two hydroxyl groups, whereas the ribose sugar of DNA has been deoxified and has only one hydroxyl group. Okay, so now we can string them together. We can make our phosphodiester bonds. We know the bases that we're going to contain, the four bases for each DNA and RNA. We know that the sequence or the chain is what matters, so the sequence of the bases is what is critically important. And so now let's talk about chains or sequences of nucleotides linked together by phosphodiester bonds. Let's represent that. Well, we can represent it this way. You see, we see our phosphate and sugar groups here kind of creating a backbone or a spine, a scaffolding. It's very repeating, phosphate, sugar, phosphate, sugar, phosphate, sugar. There's no information there. And then here are our nitrogenous bases. We have a thiamine, an adenine, a cytosine, a guanine. That's where the information is. It's that sequence, remember that sequence of those nucleotides, that sequence of those bases that really carries information. So this is a perfectly acceptable representation of a single strand of, in this case, DNA, because we're looking at thiamine. We could also represent it like this, a little bit lazier. We have our phosphate sugar, phosphate sugar, phosphate sugar backbone shown a little bit less in detail, but it's still there. And we have our bases represented by single letters, but it's the same sequence, right? TA, CG. And we can get really lazy and show the sequence like this. It carries the same information. Since the phosphate sugar backbone is so repeating and it's always there in DNA and RNA, we don't even have to show it at all. We know it's there. It's always there. It's only the bases that matter, and it's only the sequence of the bases that matter, so why not just represent that sequence as TACG? Perfectly acceptable. But there is a catch. The catch is that DNA has polarity. What does that mean? It means that direction matters. There is a beginning and an end to DNA molecules and RNA molecules. Just as in language, the direction of letters matter. GOD as a word has a very different meaning than DOG. They're the same letters in the same order, but polarity matters. Which letter comes first and which letter comes last makes a difference, and DNA is exactly the same. We always write the language of DNA from 5' to 3', even when we don't indicate it as such. So this sequence is unarguably TACG, because we write DNA from the 5' end, being the beginning, to the 3' end, being the end. From the 5' end, being the beginning, to the 3' end, at the end. So TACG. And this sequence is most assuredly not GCAT, which would be the same sequence but written in reverse. And that would be like writing a word backwards. Different meaning, or no meaning at all. The 5' end of any DNA strand is always, and then I put the caveat usually, because there are some very rare exceptions, but always has that free phosphate group. That's how it's recognized. So this is the 5' end because this is where our phosphate group is hanging out. And the 3' end has its free hydroxyl. So we always recognize the 3' end as the hydroxyl on that ribose sugar. Okay, so now what can we do? We've gone all the way to making single strands of DNA. The third difference between DNA and RNA, and the last one that we'll talk about in this lecture, is that RNA is often single-stranded. It is a single chain of nucleotides, as we've been showing and discussing all lecture here, whereas DNA is, of course, a double-stranded molecule. DNA is actually composed of two independent and distinct chains of nucleotides, each with their own phosphate sugar backbone, associating with one another. These chains go in opposite directions. It always reminds me of a highway, with one road going north to south and the other side of the highway going south to north. Two parallel roads, almost identical in their composition, almost identical in their function, nearly touching one another side by side, but one goes one way and the other goes the other way. We just talked about the polarity or direction of DNA. We talked about DNA is typically written 5' to 3'. So if we're saying that DNA strands go in opposite directions, and instead of saying north to south and south to north, we say that one strand is going 5' to 3', how would you describe the DNA that goes in the opposite direction? I'm listening very closely right now. That's right. I heard you. 3' to 5'. See, I knew you'd get it. That's why I put exactly on my slide. So for DNA, double-stranded DNA molecules, one strand is going 5' to 3' in one direction, the other strand goes 3' to 5' in the opposite direction. We use the term antiparallel to describe this. Antiparallel, shown on this diagram. The right strand is going 5' to 3' top to bottom. The left strand is going 5' to 3' bottom to top. Whoops. And DNA is not only a double-stranded molecule, it is a double helix. We all know, I'm sure, that DNA is a helical molecule. It takes on the shape of a helix. The two strands are not only associated with one another, they wind around each other. This structure was first discovered by Crick and Watson. I think at this point you all know why I don't say Watson and Crick. I have a great deal of respect and admiration for Crick, and I have a great deal of contempt and negative feelings for Jim Watson. It's a played-out analogy, but it's the one that's always used. You can think of a spiral staircase where the banisters are the phosphate sugar backbones, very, very repeating of the two strands. And the stairs connecting those two banisters, the stairs are the bases interacting in the center of the staircase. In double-stranded DNA, the bases point inward toward the center of the helix. They interact with each other, as we see here. Adenine always base pairs with thymine. And yes, in the rare occurrence that RNA is double-stranded, adenine will base pair with uracil, but let's not go there, because we're really not going into that much depth. And cytosine always base pairs with guanine, period. They base pair with one another through hydrogen bonds. Adenine and thymine happen to have 2 hydrogen bonds, while cytosine and guanine have 3. That's why you see 3 lines in the center here on the figure and only 2 lines in the center here. So GC base pairs are just a little bit stronger than AT base pairs. That's not so critically important. I'm assuming that you all have some idea what hydrogen bonds are from the chemistry courses you've taken. It's not so critical for this course, but we do need to really understand hydrogen bonds for cell and molecular biology, and I'll reteach that to you when we get there in that course. The DNA double helix is a right-handed helix. It turns clockwise. How in the world can you say that this molecule turns clockwise? Well, we can figure that out. I want you to imagine that you are shrunk down into the magic school bus on this tiny little spring here, and you're going to ride this spring down like a very, very fun slide. So we're going to start off right here on the slide, and we're going to ride the spring down like it's a long slide. And here we go, we ride it down, and whee, we're going all the way around the spring, wee, we're going all the way around, wee. What direction are we turning in? If you watch the cursor, we're turning in a clockwise direction. If you imagine this is a clock, we're turning from the 12 to the 1 around the clock. Now get up, walk over to the other ride, get to the top of that spring, and let's ride this ride down. Okay, here we go. Hold on. Whee! Whee! And look at the direction we're twisting in here. We're going in a counterclockwise direction. We're going against the clock. So indeed, DNA is a right-handed helix. It twists in a clockwise orientation from top to bottom. Those hydrogen bonds that are keeping the bases interacting in the center of the helix are weak, non-covalent bonds. They are not the sharing of electrons, but instead the sharing of a proton between a partially positive proton donor and a partially negative proton acceptor. That's what a hydrogen bond is. That is a weak force, a very weak force, non-covalent force, and that weak force and only that weak non-covalent force is all that is holding your DNA together, nothing more. Your DNA is held together by nothing more than weak non-covalent hydrogen bonds. Pretty creepy. So how is that possible? How does such a weak bond hold something as critically important and as necessary as DNA together, and hold it together so well and so tightly? It's counterintuitive. Well, the answer to that question is Velcro. If you're ever really, really bored, you should give this a try. I've tried it a number of times myself. It never gets old. Find some Velcro in some thing or another. Find some Velcro. Get a really close look at it. The way the Velcro works is that one half of the Velcro strip is all these little tiny plastic hooks. And the softer, fuzzier side of the Velcro strip is these like fabric threads that are looped. These loops of fabric thread. So what happens with the Velcro is that you push them together, and all those little plastic hooks grab onto all those little fabric loops, and that's what holds the Velcro together. So what I'd like you to do when you're really bored is go ahead and get some Velcro and try to get just one of those really tiny hooks hooked into just one of those really, really thin fabric loops. It's possible. It takes some practice. It's possible. And once you get it done, pull them apart. And you'll see it's almost like air. There's no force against you at all. It's super easy to pull them apart. Boop! And they're pulled apart. Now go ahead and repeat it, but when you repeat it, don't do just one hook and loop. Push the two Velcro strips together all the way that they go, and now try to pull those apart. And you'll see that it's much, much harder to do. Why? Well, now it's intuitive. The reason why is because you're not pulling apart one hook and one loop. You're pulling apart thousands of hooks and thousands of loops. And there is, for Velcro, strength in numbers. The same exact rationale applies for hydrogen bonds in DNA. Yeah, hydrogen bonds are weak non-covalent forces. There's not a lot of energy in a hydrogen bond. That's true. But that's true for one hydrogen bond. Now imagine multiple hydrogen bonds between every single base pair across the entire length of a single chromosome. Oh, my Lord. You're talking about lots and lots of Velcro. Lots and lots of hydrogen bonds. And all of those hydrogen bonds together have great strength and numbers and become exceedingly difficult to pull apart. So DNA is actually a very, very smart molecule without having any real intelligence or smarts whatsoever. All DNA has to know on the chemical level is that A's base pair with T's and G's base pair with C's. And that's it. You want to copy some DNA? You want to do some DNA replication? No problemo. Just pull those two original strands apart and fill in what's missing. So if you want to copy this DNA molecule into two new molecules, pull the strands apart and say, well, in the original strand, there was a C here. So what should I put across from it? Because I'm making new DNA. What should I put across from it? A G, of course, because C's always base pair with G's. And what do I put across from this C? A G. What do I put across from this T? An A. On this old strand, there was a G here. Well, I knew that. There was a G there. What do I put across from it? A C. What do I put across from this G? A C. And we wind up making two identical molecules that equal the sequence of the first, because what we're doing is we're pulling apart the two strands of DNA and using each of those original strands as a template, telling us what we should build across from it restoring those base pairs. So you've got yourself two new double-stranded molecules identical to the first, but they're not really new, are they? Each of those quote-unquote new molecules is actually one truly new strand and one old strand that came from the original acting as template. So we call DNA replication semi-conservative. What does it mean to conserve? It means to keep. When you conserve water, you keep water. When you conserve food, you keep food. When you conserve DNA, you keep DNA. So semi-conservative means we've kept some. Semi-conservative replication, replicating DNA by keeping some of what you already had, that is the one original strand, and then making new DNA, the second strand, based on that. So we are now ready for the central dogma of molecular biology. We are now ready to talk about and understand how DNA contains the information for building proteins, and briefly discuss how that information is manifest, in other words, how those proteins are made. So remember that DNA is nothing more than a storage repository, a storage device, a storage molecule for information, the information for building proteins. The proteins are the doers of the cell. The proteins are the workers of the cell. The proteins are the machines of the cell. The proteins keep us alive. So how does that information flow? How does that information get from the DNA to the proteins? It's all explained beautifully, simply, we would say in Science-ese elegantly, by the central dogma of molecular biology, which I often, but only half-kiddingly, refer to as my own personal religion. I believe in this. It is my belief. So you start off with DNA. DNA, our double-stranded molecule. We should have a very comprehensive understanding of DNA at this point. And from that DNA, you make an RNA copy. You make an RNA copy of that DNA sequence. And then you use that small, portable, Xeroxed gene in the form of RNA to translate or make your protein. So it's DNA to RNA to protein. And it's that simple. That's the flow of information. Now there's replication in there too. Some people consider that a flow of information because you're transferring or transmitting DNA information to the next generation of daughter cells. That's fine. But the central dogma really focuses on the DNA to RNA to protein flow. Information never flows backwards. So you don't go from protein to RNA. You very, very rarely go from RNA to DNA. RNA is the middleman. RNA is the intermediate between the two steps. DNA is the storage man. DNA stores the information. And the proteins are the doer mans. The proteins are built and go off and do something. So this is a touching image for me. I'm a native New Yorker, a displaced New Yorker. I was raised in Brooklyn. This is 9/11, later on in the day of 9/11, near Ground Zero, downtown, near where the World Trade Center was, but a few blocks away from World Trade Center. And what you see there is the dust on everything. The dust there is covering everything from the debris from when the towers came down. And the person on the bike is one of the omnipresent couriers, the Wall Street couriers that exist around the financial district in Manhattan. They have these backpacks, and they're always on bikes, and they're bringing information from one corporation to another, from one stock trading firm to another. They are the messengers. So the real nuts and bolts of what's going on in the financial district is really occurring between the person who is sending the message or the transaction and the person who receives it. That's the real important part. Just like the real important part of the central dogma is the DNA which stores all of the information and the proteins which do all of the actions for the cell. But where would the financial district be without the couriers? Where would the financial district be without the middlemen? How would those monies and those forms and those transaction details, how would they get in real time in a non-digital format from one person to another without the middleman carrying the message? So RNA is that middleman, equally critical to the process of the central dogma. And in fact, we call the RNA of the information flow of genes mRNA, and that M does stand for messenger, just as these Wall Street bikers are messengers. So mRNA is the messenger molecule passing the information from DNA where it is stored to the protein building apparatus so that the protein can be built. As shown in this figure, and I think I've already covered it in the course, the process of making an mRNA copy from a DNA gene is called transcription. I actually focus my own research interests on transcription. The process of making a protein from the RNA is the process of translation. And those words, those names for these processes, are not chosen arbitrarily. In fact, if you can remember what's going on, you can easily remember the name, or if you can remember the name, you can figure out what's going on. When we say transcribe, now let's get out of science for a bit. When we use the word transcribe, we mean to copy. There are transcription services of medical records. There are transcribed court records. Scribes are people who handwrite religious documents or other documents. So transcribe means to copy. To copy, faithfully copy. And indeed, a messenger RNA is nothing more than a copy of the DNA sequence it was made from. Now, there are subtle differences. There's that extra hydroxyl group on the ribose sugar. It's single-stranded, and I guess most important, there are uracils instead of thymines. Okay, fine. Great. But for all intents and purposes, DNA is a sequence of nucleotides. Messenger RNAs are sequences of nucleotides. The messenger RNA carries the same sequence as the DNA gene it was made from. This is a little animation of transcription occurring, and you can see, look at the top strand of DNA. This is the gene strand. This is the sequence we're interested in. And because of the rules of base pairing, look at the sequence we're making on the messenger RNA. CATT-CGA, CAUU-CGA. Except for the uracils substituted for the thymines, we made the same sequence. This is a small, single-stranded copy, transcribed copy of the DNA sequence. But what does the word translate mean? Well, translate means something completely different. When you translate a document, you've changed it from one language into another. You didn't just copy it. You translated or changed it from one language to another. And indeed, translation is the very same thing. We're no longer dealing with the language of nucleotides when we do translation. We are translating that DNA and RNA language of nucleotides into the language of amino acids. Amino acids are the building blocks of proteins. So this little animation shows you translation. On the bottom in red, we have the messenger RNA sequence. And as translation occurs, we are translating that sequence of nucleotides into methionine, arginine, glycine, which are amino acids. So we are going to a completely different language, a completely different chemistry of molecules when we do translation. Again, to put the fine point on it, though, the flow of information here is from the DNA gene to the messenger RNA copy to the protein chain of amino acids. That is the central dogma. So genes are runs of DNA sequences of nucleotides that carry information for building proteins. They are islands of information in the larger sea of DNA sequence that we find on the chromosome. In eukaryotes, one gene is copied into one messenger RNA and one protein is made from that. In prokaryotes, it's a little bit different. You can make many proteins from a single gene in prokaryotes, and we'll talk about that in 2 lectures. We'll leave that there for now. Either way, though, regardless of your cell type, proteins are being made using the information contained within the sequence of nucleotides first in the gene and then copied onto the messenger RNA. The messenger RNA was a direct copy of the DNA sequence. That means transcription is an easy process to carry out from the cell's perspective and an easy process to understand. I mean, God, my 5-year-old daughter could transcribe a DNA sequence for us if we wanted her to. A monkey could probably be trained to do this. When you see a G, put a C across from it. When you see a U, put an A across from it. That's transcription. Translation, not so straightforward. How do we translate that language of nucleotides into protein? So DNA is chains of nucleic acids. RNA is chains of nucleic acids. Messenger RNAs are copied chains of nucleic acids from the DNA, still following those rules of base pairing. But proteins in their simplest form are chains or strings of amino acids, So how do we do that conversion? How do we translate a chain of nucleotides into a chain of amino acids? If we can do that, we will have completed the central dogma and completed the flow of information. If we can't, then we stop before the proteins are built, and we've done it all for nothing. So let's talk about building proteins. There are 20 amino acids that are used by cells to build proteins. Now you can use the analogy of letters here too. These amino acids are the letters of proteins. We have 26 letters in our English alphabet. There are 4 letters in the alphabet of nucleotides, and there are 20 letters, or 20 different types, in the alphabet of amino acids. Again, in their simplest structure, proteins are nothing more than chains of these amino acids strung together, just as DNA was chains of nucleic acids or nucleotides strung together. There are 4 different flavors of DNA. We've already covered this. A, T, G, and C. And these 4 different nucleotides have to, in some way, encode or be translatable into the 20 different amino acids. So we can't just have a simple system where one DNA base codes for one amino acid. Well, maybe we can. Let's try. 20 amino acids, 4 bases. A, every time you see an A, put in that place for the protein amino acid number 1. Fine. So far, so good. T, every time you see a T in a messenger RNA, put in that place amino acid number 2 in the protein you're building. Well, maybe this will work. C, every time you see a cytosine in a messenger RNA, oh, I should have said uracil above, sorry about that. Every time you see a cytosine in a messenger RNA, put amino acid number 3 in the protein you're building from it. G, every time you see a guanine in your messenger RNA, put amino acid number 4 on it. You want to add amino acid number 5? Tough noogies. We're out of nucleotides. So that's not going to work. We need some way where nucleotide sequences carry the information for amino acid placement. Let's try 2. Let's say 2 DNA nucleotides, and therefore RNA nucleotides once we've done transcription. Let's say 2 bases codes for or carries the information for a single amino acid. Well, if we do that, we're actually only good for 16. We've got 4 bases in the first position. We've got 4 bases we can put in the second position. 4 times 4 is 16. Still not enough. We've got 20 amino acids. We have to have at least 20 different possible combinations of nucleotides. So let's try 3. How about 3 bases? Well, if we use 3 DNA bases, then it's 4 times 4 times 4, which is 64. We only needed 20. So this is enough. In fact, it's more than enough. But believe it or not, more than enough is a good thing. There are 64 different possible combinations or sets of 3 DNA bases. What we mean by combinations is you can have AAA, you can have ATA, you can have AGG, you can have AGA. There's 64 of those, and we call them triplets because they are groups of 3. We also refer to them as codons because these 3 nucleotides code for one amino acid. And again, having more than 20, having 64 is a good thing. DNA can be mutated. That's a fact of life. In fact, the next lecture that we're going to talk about after this one talks all about DNA mutations. You can't avoid it. You can't avoid it. If DNA's only job is to provide the instructions for building proteins, then a mutation in that DNA is only a bad mutation if it changes the protein we make. Does that make any sense? Think about that. It's all about the protein. You make the right protein. I don't care how many mutations you got. You make the right protein the right way, it's like you didn't have a mutation at all because that was the DNA's job in the first place. If you change the instructions, but the protein that's made in the end is the same exact protein, that's not a bad mutation. Consider this example. Here's some instructions for you. To get dust off of your hat, beat it against the wall before putting it on your head. To get dust off of your hat, beat it against the wall before putting it on your head. That's your instructions. That's your DNA sequence. Now, let's mutate that. So let's mutate the US of the word dust into IR. That's a 2-base, 2-letter mutation. To get dirt off your hat, beat it against the wall before putting it on your head. Works for me. I mean, it's dust in the first sentence and dirt in the other. Did we change the sentence? Yeah. Was that a mutation? Yeah. But in the end, it's kind of the same meaning, right? Same instructions, really, no? So that's not a bad mutation. That's an okay mutation. Let's see what a bad mutation is. Now, a single letter change. To get dust off your cat, beat it against the wall before putting it on your head. Well, gee, just one letter. We actually changed fewer letters than we did in the first mutation, but that's not a good mutation. That's not an okay mutation. That's a bad mutation. It's a pretty different story there, no? By changing that single letter, we change the entire meaning of the sentence a lot. And we're most certainly going to change the end result of these instructions. We're going to have a really pissed off cat that didn't like being beat against the wall, and we're going to be putting that cat on our head. So we have different categories of mutations, some that are tolerable and some that are really bad, depending on how that mutation affects our instruction manual. Having 64 codons for 20 and only 20 amino acids actually allows for okay mutations. We call these okay, tolerable mutations, silent mutations, because we don't even know they're there. Here's the genetic code. You can think of this table as your nucleotide to amino acid dictionary. Just as we might have a Spanish to English dictionary, this is your nucleotide to amino acid dictionary. All in triplets, because triplets of nucleotides carry the information for a single amino acid. So you want to decipher what CGG means in amino acid language. The triplet or codon CGG in the nucleotide language translates to arginine, the amino acid arginine in the amino acid language. Now let's talk about some good or silent mutations. So in my genome, I have a gene, and that gene has a code in it. That code is CGC. The instruction manual says, you put arginine in this spot, CGC. And then I pick up a mutation, and heaven forbid I give that mutation to my kid. I pass that mutation on to my child. I give them a mutated gene. And the mutation I gave them is that this C here in this third position mutated to an A. So now instead of CGC arginine, I tell my unfortunate child's gene to make CGA arginine. Well, in fact, I didn't really change anything at all. It's all about the protein being built. The protein needs arginine in that position. My non-mutated gene calls for arginine in that position. My daughter's mutated gene calls for arginine in that position. No harm, no foul. 6 of one, half a dozen of the other. And any other cliche you can come up with that states that no damage was done. This is called redundancy. And having 64 codons for 20 amino acids allows you to have multiple codons code for the same amino acid. So if you pick up any of those mutations, you have a silent mutation. You don't see the effects at all. All proteins start with an AUG in RNA or an ATG in DNA codon. That is the start codon that initiates or begins this translation process. AUG codes for the amino acid methionine. And so all proteins begin with an AUG start codon. That's how we recognize protein coding genes. All genes start that way. And then we just simply take triplets, 3 at a time, starting with the start codon. After we see the AUG start codon, methionine goes in that position, then the cell looks and says, what are the next 3 nucleotides? Well, if the next 3 nucleotides are CCA, then we put a proline in the next position for the protein. And then we look at the next amino acid, the next triplet. And the next triplet is GCG. Well, that's alanine. So we put alanine in that position. So we take the nucleotides 3 at a time, all the way down until we find and encounter a stop codon. UAA, UAG, or UGA. And these 3 codons don't code for amino acids. They are calls to the translation machinery, the translation proteins, to stop this translation. The protein is done. It's like the return key on a word processor. Line is finished. We call the process of stopping translation translational termination for obvious reasons. And so, stated most simply, cellular machinery, the ribosome, in fact, we'll get to that on the next slide, lands at the very beginning or 5' end of a messenger RNA and just starts scanning, moving down that messenger RNA for the first AUG start codon. Right at that position, a methionine is placed on the beginning of the protein, and then we start adding amino acids by reading the codons 3 at a time, and we start building a chain of amino acids until we finally reach a stop codon. That is the process of translation. So what's doing all this? What's doing the scanning? What's landing on the RNA? What's making the protein? Ribosomes. The ribosome is the site of translation. The ribosome allows the information contained within the messenger RNA, which came from where? The DNA gene originally, to be used to create a chain of amino acids. Ribosomes, whether they're in our own cells or bacterial cells, are huge, huge. They are protein and RNA conglomerates made up of well over 60 different proteins and RNA molecules. They are large. The ribosome lands on that 5' end of the messenger RNA. It does the scanning. It recognizes the start codon, and it sees to the building of a protein. It doesn't do it alone. Many, many other factors that are needed, but the ribosome is really the coordinator of this process. So the ribosome keeps adding amino acids as they are called for by the triplets or codons until that stop codon is encountered. The deeper question, well, how does this work? How do you add amino acids? How do you know where to find the amino acids? How does the ribosome do all this? Well, if you're wondering all of those things, hold on to those questions for about 4 more months. And if you take Bio 320, Cell and Molecular Biology in the spring, we will answer all of those questions quite thoroughly. Last slide on protein folding, very, very quickly. So translation is done. What do you have to show for it? You've got a chain of amino acids. You started with a methionine, you end with a stop codon, and in the middle you have hundreds, if not thousands, of amino acids strung together in a chain. So what happens now? Well, spontaneously, for biochemical reasons, which you will cover in your biochemistry course, this linear chain of amino acids folds up into 3-dimensional shapes. First, it spontaneously folds into what's called secondary structures. Then those secondary structures kind of knot and loop together to form what are called tertiary structures. And then sometimes multiple proteins actually come together and form large multi-protein structures that we call quaternary structures. But this is your active protein. Proteins do the jobs they do and have the functions they have because of the 3-dimensional shapes that they fold into. The 3-dimensional shape of a protein allows it to do its job. That protein folds into that 3-dimensional shape as a direct result of its amino acid sequence. And that amino acid sequence was dictated by the sequence of nucleotides in the gene that encoded it. So the gene encodes the sequence of amino acids. That sequence of amino acids dictates the folding. The folding dictates the function, and it's the function that's keeping us alive. DNA is, quite literally, the blueprints of life. So the grand goal of this lecture was to refocus our attention now on DNA no longer as a molecule of inheritance, but a physical molecule of function. We need to understand how the DNA molecule is made, looks, behaves, and functions as an information repository if we are to move forward in this course. So we started off just talking about what DNA does abstractly. DNA is the encyclopedia of the cell, holding all of the information for building all of the proteins that the cell may need. RNA, then, is that single Xeroxed page from the encyclopedia that you need at this moment at this time. RNA is smaller, portable, and only for specific genes. We talked about nucleotides. Nucleotides have 3 things in common with one another. They all have their phosphate groups. They all have their sugar group, that ribose sugar, and they all have a base, but it's the base that differs. There are 4 different bases that we are concerned with: guanine, cytosine, adenine, and thiamine or uracil, depending on if we're talking about DNA or RNA, respectively. These nucleotides are held together into sequences or chains through phosphodiester bonds, and it is the sequence that carries the information. DNA has polarity. The direction of DNA matters, and we always consider DNA from its 5' end at the beginning to its 3' end at the end. DNA is also double-stranded, and it is anti-parallel, with one strand going in one direction and the other strand going in the other. We covered the rules of base pairing. Thymine always base pairs with adenine always base pairs with thiamine, and cytosine always base pairs with guanine through hydrogen bonds. We talked about how these rules of base pairing lead to semi-conservative replication. By separating the 2 strands of an existing DNA molecule, you can use each of those single strands as a template strand and fill in the complementary base on the other side. So each quote-unquote new molecule of DNA is actually composed of one old strand and one new strand. That led us straight into the central dogma. We just started by saying the flow of information for the central dogma is from the gene on the DNA, copied faithfully to a nucleotide sequence that matches on the messenger RNA, and then translated into a completely different language of amino acids, building a protein. That means RNA is the middleman. The ribosome is what sees to protein synthesis, building the protein off of that middleman messenger RNA. Messenger RNAs are scanned by ribosomes starting at the 5' end, looking for that first all-important start codon. That marks the addition of a methionine in that position for the protein. And then we take triplets, codons, 3 nucleotides at a time, reading down this messenger RNA, decoding it, putting amino acids in a chain as they are called for by the triplets, using that genetic code, that nucleotide to amino acid translation dictionary. I dropped the stuff on tRNAs. I'm sorry it's here on the summary slide. We're not responsible for tRNAs. And then we ended the lecture by talking extremely briefly about protein folding. It's not enough just to make a chain of amino acids. That chain of amino acids then folds in a stepwise fashion into a 3-dimensional shape, and it's the 3-dimensional shape of the protein which actually allows it to do the job that it does. Now, with all of this in hand, and all of this in mind, we have a much better context by which to describe gene mutations. So we're going to go back to textbook material, back to a little bit more of a structured, formatted lecture style when we regroup on the next lecture. That'll be chapter 18 from your text, talking about gene mutations and DNA repair, how mutations can be fixed if they're spotted soon enough. As is always the case, thanks for watching.